And while it might seem an unlikely topic for a reservoirs meeting, I would hope that you would consider the various cellular factors that a virus must encounter on its way to establish a productive infection and therefore establish viral reservoirs. So to begin with, in the search for developing a suitable non-human primate model for HIV-1 transmission, there was initial frustration with the inability to infect rhesus macaques with HIV-1. This frustration turned into excitement in 2004 when it was discovered that the main reason for this block to infection is rhesus TRIM5 alpha. Now, TRIM5 alpha is a protein that various species express, and their different variants are active against different viruses, which you see in the table below. And importantly to all of us here, rhesus TRIM5 alpha is very potently active against HIV-1. So to go into a little bit about how we know TRIM5 alpha blocks HIV-1 infection, we know that it exerts its effects very early after entry into the host cell. And if you look in this cartoon here, you will see that I've represented TRIM5 alpha as these green dots. This is because when we express TRIM5 alpha exogenously in cells, it localizes to what is known in the field as cytoplasmic bodies, just accumulations throughout the cytoplasm. And unfortunately, there's no antibody that recognizes endogenous TRIM5 alpha, so this is all we know of its expression pattern. So we know that after the virus capsid core is deposited into the host cell cytoplasm, the TRIM5 alpha recognizes the capsid, and this normally results in a failure of the virus to complete reverse transcription. We know this by using quantitative PCR looking at late reverse transcription products. If you look in the left panel, the first pair of bars is in red, cells expressing TRIM5 alpha, and in yellow are just plain HeLa cells, and you can see that reverse transcription is very potently blocked. If you look on the graph on the right, you can see that there is a correlation with a block to infection. However, if you carry out infection in the presence of MG132, which inhibits the proteasome, despite infection being blocked, which you see on the right, we now see that reverse transcription is able to fully recover. So this led to the current working model we have of the mechanism of restriction. First, TRIM5 alpha will bind to the capsid core, and then this is followed by a normal block of reverse transcription, which occurs in a proteasome-dependent manner. However, if you conduct infection in the presence of a proteasome inhibitor, such as MG132, you can see this intermediate stage of restriction, which is the virus is able to reverse transcribe. However, this pre-integration complex is unable to access the nucleus and therefore unable to establish a productive infection. Our lab has previously been able to visualize this intermediate stage of restriction. In this figure here, you will see the virus, which is labeled with GFP-VPR, appears to be sequestered inside of a TRIM5 alpha cytoplasmic body, and this is in the presence of proteasome inhibition. Furthermore, TRIM5 alpha cytoplasmic bodies have been shown to contain ubiquitin. So the fact that proteasome inhibition leads to this effect on reverse transcription, and additionally that ubiquitin does appear to be localized to these cytoplasmic bodies, leads to the question of what is the role of the proteasome? Is the ubiquitin that's localizing to cytoplasmic bodies signaling for the proteasome to come in and degrade a component of these cytoplasmic bodies, whether it be TRIM5 alpha, the capsid, or some other protein that localizes to these areas? To begin to explore this question, we took an imaging approach using a fluorescent proteasome construct called LMP2-GFP. Now, LMP2 is a beta subunit of the proteasome, and this construct has been extensively characterized as representing functional proteasomes. So what I first wanted to look at was whether the proteasome is even poised to be able to have some impact on components of cytoplasmic bodies. So what you see here is that I've transfected LMP2-GFP into cells that express an HA-tagged version of TRIM5 alpha, and I see that proteasomes predominantly localize diffusely throughout the nucleus. I do see some signal in the cytoplasm, but it is not nearly as much as what I see when I inhibit the proteasome with MG132. Now in this case, I see that proteasomes have extensively relocalized out into the cytoplasm and overlap very nicely with the expression of TRIM5 alpha. Now, proteasome inhibition is not the most physiologically relevant scenario, and what is even more interesting to me is that when I treat with virus alone without proteasome inhibition, I see the same effect. I see TRIM5 alpha forming their typical enlarged cytoplasmic bodies, which others have shown occurs with proteasome inhibition, but I also see that proteasomes localize to these cytoplasmic bodies. So having seen that proteasomes can localize to TRIM5 alpha cytoplasmic bodies, and especially that this does happen with the presence of virus, the next question was whether virus might be able to interact with the proteasomes in these TRIM5 alpha cytoplasmic bodies. To answer this question, I perform some live cell imaging. And what you'll see in the next few movies, in green is going to be proteasomes with LMP2-GFP, and in red is the virus, which is tagged with mCherry-VPR. And zooming in on this movie here, you can see that yes, indeed, they do associate in living cells. I don't know that I know how to play the movie. Let me see if I can work this. Okay. Oh, no. How do I click? I need to click on the movie. Oh no, go back. Can you click back there? It's this slide, yeah. This one? Yeah. Well, it's also on some Oh, that's okay. I checked it beforehand. That's unfortunate. Well, what you were supposed to see in this movie is that the proteasomes and also the virus are associated with each other, and that they are both moving dynamically throughout the cell, and they remain associated for the duration of this movie, and most of these movies are for 30 minutes. Oh, that was actually the wrong one. Okay, so that was what you're supposed to see in this movie. So then I also see a similar type of interaction, which is a long-term stable association that is also characterized with a decrease in the associated virus's signal that is independent of general effects of photobleaching. And we can't watch these movies either, but we can see the time lapse here. And if you look in the green and in the red, you can see that the virus and proteasome remain associated, in this case, for about 20 minutes. And the virus does appear to be decreasing its fluorescence during the course of this association. So quantifying these associations, in addition to the stable interaction that you didn't actually see that I described first, and the stable interaction associated with a decrease in virus signal, I also see what I call escape, which is when the virus and a puncta of proteasomes appear to traffic with each other for at least several minutes, but the virus is able to leave this proteasome puncta. And additionally, I rarely see a proteasome puncta associated with the virus, but the punctum loses its punctate localization, becomes more diffuse. So what you see in this graph here is I've quantified the proportion of association events that I see either with virus alone or with virus with proteasome inhibition. And what I do notice is I see, for example, more escape events when proteasome inhibition is added with the virus, and I see more stable interactions when there is no proteasome inhibition, which is interesting in that stable interactions appear to, if not require, at least benefit from a functional proteasome, which might be indicating mechanistically that the proteasome might actually be turning over some component of the cytoplasmic bodies, perhaps the virus. So these studies are ongoing. As I've shown you, or tried to show you without the movies, proteasomes and virus do associate within living cells, and previous work from our lab has shown that virus associates with TRIM5 alpha in living cells in a similar manner. It would also be interesting to look at the interactions of proteasomes and TRIM5 alpha in living cells. And additionally, I see that proteasomes are recruited. Now, if they are recruited in order to degrade some component of cytoplasmic bodies, it would make sense that ubiquitin is brought in first. And while we know ubiquitin localizes to the bodies, it would be interesting to monitor the dynamics of these components of the ubiquitin proteasome system being brought in. Additionally, the live cell imaging is all using a construct based on one subunit of the proteasome, but it would be interesting to look at which subunits of the proteasome are recruited. Proteasomes are composed of the 20S catalytic core as well as many 19S regulatory subunits. And there's just a couple figures here. On the top is a regulatory subunit called RPN10, and you can see that this one also does localize to TRIM5 alpha cytoplasmic bodies, and whereas the subunit below RPT5 does not have this specific localization. So in conclusion then, proteasome localization with regard to TRIM5 alpha especially appears to be dynamic. Proteasomes are able to localize to rhesus TRIM5 alpha containing cytoplasmic bodies, and this can be induced either by virus or by proteasome inhibition. I've also shown you that virus can associate with proteasomes in living cells with several characteristic types of association, and that there does appear to be a difference whether you have a functional proteasome or whether you inhibit it. And in general, then this highlights the importance of various cellular factors and the ability of the virus to establish a productive infection. I'd like to thank the HOPE Lab and those who've provided reagents and funding, and I'd be happy to take any questions. Thank you.
Thank you very much. We're all sorry that the movie didn't work because it's always such a treat to see such beautiful images. So are there questions from the floor?
Yeah, I'm sorry if I missed it. Maybe you showed it. Do you see ubiquitin at all? And if yes, do you have any idea about the linkage?
Yeah, I didn't have time to show you, but I do see, I've stained with antibodies specific for either conjugated only ubiquitin or polyubiquitin conjugated ubiquitin. The slide I've shown you previously would recognize either free or conjugated ubiquitin. I do see that there is mono and polyubiquitination occurring in these TRIM5 alpha cytoplasmic bodies. Was that not? Yeah, so I've looked with a K63 specific antibody, and I did not see much there. Unfortunately, I cannot get a hold of a K48 one. The last I knew, it was not commercially available. And yeah, no, but that's definitely interesting to look at what type is there and whether it is the one that would recruit proteasomes. I'm certainly looking at that.
Any other questions?